All right, guys, you should be able to hear me now. Hopefully. I've been at it for an hour. I'm so bad with technology. Um, so let's get it going in the next minute. I'm just gonna confirm that there's actually noise coming out. Today we'll talk about gas exchange and carb metabolism regulation. Okay. So ChatGPT should be with us too. So let's see what our friends doing. ChatGPT, are you here?
Yep, I hear you. Sounding better now?
Hopefully. Guys, can you actually hear ChatGPT?
Testing, testing. How's it sounding, everyone? Great. Let's get started.
All right, let's start with our first week. Guys, I know I kind of let you down twice already because it takes me time to fix this, but yeah, let me know you can hear. ChatGPT, can we get started with what gas exchange is, gas transport?
Sure, gas exchange or gas transport is how oxygen gets into your blood and carbon dioxide is removed. It happens in the lungs where oxygen enters the blood and CO2 is expelled.
Interesting. Perfect. And let's talk a bit about carb metabolism too. I know, guys, these
Sure.
two. I know these two aren't very related, but I like multitasking. What can I say?
Hey, multitasking's great. So, carbon metabolism involves how your body uses carbon containing molecules, like glucose, for energy. It's essential for producing ATP, which fuels your cells.
Okay, so I think we should kick off with the regulation of carbohydrate metabolism. So why is this important? Why should we care about it?
Carbohydrate metabolism is crucial because it's how our bodies convert carbs into energy. Proper regulation ensures stable blood sugar levels, which is vital for energy, brain function, and overall health.
Can you give us a general understanding of glycolysis then, and tell us all the different weird molecules that we need to know, acetyl-CoA, PFK1, and all of this?
Absolutely. Glycolysis is the process where glucose is broken down into pyruvate, producing ATP. Key players include glucose, pyruvate, ATP, NADH, and enzymes like PFK1, which regulate the process. Acetyl-CoA is the next step, leading into the citric acid cycle.
All right, so can you build for us kind of a mind map almost?
Sure, picture glycolysis as a pathway. Glucose enters, gets converted through a series of steps into pyruvate, producing ATP and NADH. PFK1 controls a key step. Pyruvate then converts to acetyl-CoA, which enters the citric acid cycle. Think of each step as a station along the way.
Interesting. Okay. Let's talk about the glycolytic pathway. Is it the same thing as glycolysis?
Yes, the glycolytic pathway is another name for glycolysis. It describes the same series of reactions where glucose is broken down to produce energy.
Guys, let me know if you can hear well, by the way.
Just checking in. How's the audio for everyone?
ChatGPT is checking out too. All right. Okay, so sorry, let's go back. I didn't concentrate. Is glycolysis the same as glycolytic pathway then?
No worries. Yep, glycolysis and the glycolytic pathway are the same. Both terms describe the process of breaking down glucose to produce energy.
Okay, so they're the same. So we have Dude, it's so confusing. I mean, we have to spend some time understanding this. So we have glucose that becomes G6P and then F6P, and what's all this?
It can be a lot. So glucose first converts to G6P, which stands for glucose 6 phosphate. Then it changes to F6P, or fructose 6 phosphate. These are intermediate steps, preparing glucose for further breakdown into energy.
Okay, interesting. Um, can you? Okay, so we go from glucose to G6P to S6P. Then what happens after that?
After F6P, it converts to F16BP, which is fructose 1,6 bisphosphate. This molecule then splits into two 3-carbon molecules, which continue through glycolysis to produce pyruvate, ATP, and NADH.
Okay. Um, Interesting. So what's oxaloacetate?
So hexokinase is an enzyme that catalyzes the first step, turning glucose into G6P. It essentially traps glucose inside a cell so it can be used for energy.
What about oxaloacetate?
Oxaloacetate is a 4-carbon molecule involved in the citric acid cycle. It combines with acetyl-CoA to form citrate, starting the cycle that produces energy.
Interesting. Okay. Um,
Glad it's making sense. Anything else you'd like to explore?
so gluconeogenesis is not a simple reversal of glycolysis. Why?
Exactly. Gluconeogenesis isn't a simple reverse because some glycolysis steps are irreversible. It uses different enzymes and pathways to bypass these steps, ensuring the process can run smoothly in the opposite direction.
Can you elaborate?
Sure. In glycolysis, enzymes like hexokinase and PFK1 catalyze irreversible steps. Gluconeogenesis bypasses these using different enzymes like glucose 6 phosphatase and fructose 1,6 bisphosphatase.
Okay, dude, can
enabling
you
glucose
can you
production.
speak a bit slower, please?
Of course. Gluconeogenesis uses different.
Yeah, slower, but you don't need to like change your voice and make it soothing.
Got it. Gluconeogenesis uses different enzymes to bypass glycolysis's irreversible steps, allowing glucose production.
Okay, interesting. Okay. So what are the requirements for gluconeogenesis?
The main requirements are substrates like lactate, glycerol, and amino acids, along with energy from ATP and GTP. It also needs specific enzymes to bypass glycolysis steps.
Lactate, glycerol, and amino acids.
Exactly. These are the building blocks. The liver primarily uses them to synthesize glucose, especially during fasting or intense exercise.
Okay. And the urea cycle is different than the citric acid cycle or whatever it's called?
Yes, they're different. The urea cycle detoxifies ammonia into urea, while the citric acid cycle generates energy from acetyl-CoA. They occur in different cellular compartments and have distinct functions.
Interesting. Okay. So how does the urea cycle connect with gluconeogenesis?
They connect through amino acid metabolism. When amino acids are used for gluconeogenesis, their nitrogen is converted to urea in the urea cycle. This helps safely remove excess nitrogen from the body.
Okay. Interesting. So can we elaborate on that?
Certainly, during gluconeogenesis, amino acids are deaminated, releasing nitrogen. This nitrogen enters the urea cycle, where it's converted to urea and excreted, preventing toxic ammonia buildup. This process links amino acid breakdown to gluconeogenesis and the urea cycle.
What does it mean that to use amino acids as a source of carbon skeletons for glucose production, we have to first transaminate them to lose their ammonia. I don't understand why we need to do this.
Transamination is needed to remove the amino group from amino acids, leaving behind the carbon skeleton. This skeleton is then used in gluconeogenesis to produce glucose. The ammonia that's removed is processed in the urea cycle to prevent toxicity.
Interesting. Okay. So ammonia is toxic to cells, so we don't want it in the body.
Exactly. Ammonia is harmful because it disrupts cellular processes. The body converts it to urea, which is much less toxic, and then excretes it through urine.
Interesting. So so ammonia, how does ammonia get converted to urea in the liver?
In the liver, ammonia enters the urea cycle. It combines with carbon dioxide to form urea through a series of enzymatic steps. The resulting urea is then transported to the kidneys for excretion.
Okay, interesting. So so okay. So what's this equation? Enlighten me. We have ammonia with CO2 and water and ATP and aspartate, which creates urea, fumarate and ADP and AMP. Like this is complete abstraction to me. You have to kind of help me out here.
Sure. That equation represents the overall urea cycle. Ammonia, CO2, and aspartate combine, using energy from ATP to produce urea, which is excreted, and byproducts like fumarate, ADP, and AMP. Each step is catalyzed by specific enzymes.
Tell me again.
Of course, the urea cycle transforms toxic ammonia into urea using CO2, aspartate, and ATP. Each reaction is driven by enzymes, resulting in urea and other byproducts like fumarate, which are safely handled by the body.
Okay, so in the gluconeogenic pathway, so we have so many things. So I mean, I have this complicated diagram and I don't know where to start. We have fructose 1,6-biphosphatase that goes to fructose 6-biphosphatase, which will go to glucose 6 phosphatase, which will go to glucose, which I guess is kind of the opposite of what happens, right?
Exactly. In gluconeogenesis, fructose 1,6-bisphosphatase converts F16BP to F6B, and glucose 6-phosphatase converts G6E to glucose. These steps reverse glycolysis, allowing glucose production.
And then we have DHAP and GLY3P and 1,3-DPG and 3PG. What is all of this?
These are intermediates in both glycolysis and gluconeogenesis. DHAP and G3P are involved in energy storage and release. 1,3-BPG and 3PG are steps in the conversion of these molecules during the pathways.
Okay, let's talk about just a sec. So let's talk about PFK1. Why is it subject to energy dependent allosteric regulation by ATP, AMP, and hydrogen ions?
PFK1 is a key glycolysis enzyme. It's regulated by ATP, AMP, and hydrogen ions because they signal the cell's energy state. High ATP inhibits PFK1, slowing glycolysis, while AMP and hydrogen ions activate it, speeding up energy production.
Okay, okay. So ATP inhibits and then AMP activates.
Exactly. ATP signals there's plenty of energy, so it inhibits PFK1. AMP indicates low energy, so it activates PFK1 to speed up glycolysis.
Interesting. Okay. And then when hydrogen ions increase, during anoxia or anaerobic muscle contraction as a result of lactic acid production? What happens out of the two?
When hydrogen ions increase, indicating more lactic acid, they activate PFK1. This speeds up glycolysis to produce more ATP, helping muscles continue contracting despite the acidic environment.
Wait, so increase in hydrogen ions, you said, inhibits glycolysis?
Actually, high hydrogen ions can inhibit PFK1, slowing glycolysis to prevent excessive acid buildup. However, other factors can still drive energy production.
So why does increase in H plus inhibits glycolysis?
The increase in hydrogen ions lowers pH, creating an acidic environment. This inhibits PFK1 to prevent excessive glycolysis, protecting cells from damage due to too much acid.
So what does PFK1 stand for?
PFK1 stands for phosphofructokinase 1, an enzyme that regulates a key step in glycolysis.
Which step are we
It
talking
controls.
about?
That's the step where fructose 6-phosphate is converted to fructose 1,6-bisphosphate, a key regulatory point in glycolysis.
So it's when glucose 6-phosphate becomes fructose 1,6-biphosphate.
Exactly. PFK1 catalyzes that conversion, making it a crucial control point in glycolysis.
Interesting. So, okay, so can we explain to me regulation of PFK1 by nutrients?
Sure. PFK1 is regulated by nutrient levels. High ATP or citrate inhibits it, signaling enough energy. Low energy, indicated by high AMP, activates PFK1, boosting glycolysis. This ensures energy production matches the cell's needs.
So fructose 6-phosphatase, am I saying it right? So what does it mean, sign of high rates of glucose entry or glycogen breakdown?
Fructose 6-phosphate indicates high glucose availability or glycogen breakdown, signaling the cell to proceed with glycolysis. It helps the cell manage energy production efficiently based on available resources.
Why does it indicate this? I don't understand.
It indicates this because fructose is a product of glucose entering glycolysis or glycogen breakdown. High levels mean there's plenty of substrate for glycolysis, signaling the cell to continue producing energy.
And why does, okay, let's talk about fructose 2,6-biphosphate. How does it differ from fructose 6-phosphate then?
Fructose 2,6-bisphosphate is a potent activator of PFK1, enhancing glycolysis. It's different from fructose 6-phosphate, which is just an intermediate. Fructose 2,6-bisphosphate ensures glycolysis continues even when energy levels are high.
So why is it such a potent allosteric activator?
It strongly enhances PFK1's activity, overriding inhibitory signals like high ATP. This ensures glycolysis can proceed when needed, especially under hormonal regulation.
Dude, I have no idea what we're talking about. I'm sorry. Let's kind of go a step back and so I can understand the regulation of PFK1. So fructose 6-phosphate and fructose 2,6-biphosphate regulate PFK1, but I don't understand because I thought that, okay, yeah, explain to me. Because they're very similar to glucose 6-phosphate and fructose 1,6-biphosphate that PFK1 kind of transforms. I don't understand.
No worries. Fructose 6-phosphate is a normal glycolysis intermediate. Fructose 2,6-bisphosphate, made by a different enzyme, acts as a powerful activator of PFK1, ensuring glycolysis continues even if ATP is high. It's like an extra boost for glycolysis.
Why though?
Fructose 2,6-bisphosphate ensures glycolysis can respond quickly to hormonal signals like insulin. It's a way for the cell to fine-tune energy production even when other signals might slow it down.
How does it ensure this?
It binds to PFK1, increasing its activity even when ATP levels are high. This helps keep glycolysis running efficiently under different conditions.
Okay. And citrate inhibits? I don't understand. Signals TCA cycle overload. What's all this about? Or fatty acid oxidation?
Citrate signals the cell has enough energy from the TCA cycle or fatty acid oxidation. It inhibits PFK1, slowing glycolysis to prevent excess energy production.
Can you explain this a bit more to me?
Sure, citrate is a signal that the cell's energy needs are met. When citrate levels are high, it tells PFK1 to slow down glycolysis, avoiding unnecessary glucose breakdown.
Dude, I'll be honest with you. So, I don't understand.
No problem. It's a lot to take in. Want to break it down step by step?
Yeah, I don't understand why citrate inhibits and why signals TCA cycle overload or fatty acid oxidation.
Citrate indicates that the TCA cycle is producing plenty of energy. When there's excess citrate, it signals that the cell doesn't need more glycolysis, so it inhibits PFK1.
I don't understand glycolysis. I don't understand why G6P becomes F6P anyway. What exactly changes?
In that step, a phosphate group is added to fructose 6-phosphate, forming fructose 1,6-bisphosphate. This makes the molecule more reactive, allowing glycolysis to proceed.
Okay. And explain to me, so PFK2 goes to fructose 2,6 biphosphate and then fructose 2,6 biphosphatase makes, what's all that about?
PFK2 produces fructose 2,6-bisphosphate, which activates glycolysis. The enzyme fructose 2,6-bisphosphatase breaks it down, reducing glycolysis. It's a balance to fine-tune energy production.
Okay, and then so what's DHAP?
DHAP, or dihydroxyacetone phosphate, is an intermediate in glycolysis and gluconeogenesis. It's formed from fructose 1,6-bisphosphate and can be converted to glyceraldehyde 3-phosphate for energy production.
Okay, and how is this part of glycolysis? What's its function?
In glycolysis, DHAP is converted to G3P, ensuring that both molecules continue down the pathway for ATP production. This conversion helps balance the energy yield.
So, okay, let's talk about fructose 2,6 biphosphate a bit more. So, it's synthesized from fructose 6 phosphate.
Yes, fructose 2,6-bisphosphate is synthesized from fructose 6-phosphate by PFK2. It's a potent activator of PFK1, boosting glycolysis when needed.
And why does it need ATP for this?
ATP provides the phosphate group for the reaction, converting fructose 6-phosphate into fructose 2,6-bisphosphate.
Oh, I
This phosphorylation
see.
step is essential.
That makes sense.
Glad that clicked. Anything else you'd like to dive into?
Wow, this is so confusing, bro.
It can be. We can slow down and tackle each part step by step.
Um, so let me think what I'm gonna. It's kind of making more sense. Um, let's talk
That's
about
great.
the activation of gluconeogenesis. So explain to me, why does increased fatty acid oxidation lead to increase in acetyl-CoA?
Sure. Fatty acid oxidation breaks down fats into acetyl-CoA, a key molecule in energy production. More fatty acid oxidation means more acetyl-CoA, which can then enter the TCA cycle for energy.
Okay, interesting. So can you explain this a bit more?
Sure. When fats are oxidized, they produce acetyl-CoA. This enters the TCA cycle, generating energy and signaling that the cell's energy needs are met, which can reduce the need for gluconeogenesis.
And how does it give us the needs that we need? What acetyl-CoA, how does it give us energy?
Acetyl-CoA enters the TCA cycle, where it's broken down to produce ATP, the cell's main energy currency. This process provides the energy needed for various cellular activities.
Dude, I'm confused.
Yeah, it's a lot. We can keep breaking it down if that helps.
Please, let's do that. Yeah.
Sure. So acetyl-CoA from fatty acid oxidation fuels the TCA cycle, producing ATP. This process reduces the cell's need to make glucose through gluconeogenesis.
One more time.
So fats break down into acetyl-CoA, which enters the TCA cycle, producing ATP. This energy production signals the cell that there's enough fuel, so less glucose needs to be made.
All right. And why does, okay, and we have increased glucagon. So why do we have increased glucagon? Why does increased glucagon inhibit PFK2 activity and stimulate F26 biphosphorylation? I don't understand all these complicated words.
No worries. Glucagon signals low blood sugar. It inhibits PFK2, reducing fructose 2,6 bisphosphate, which slows glycolysis. It also activates fructose 2,6 bisphosphatase, further lowering fructose 2,6 bisphosphate
Okay,
and favoring.
if you can speak a bit slower and louder. So explain to me how glucagon inhibits PFK2 activity first, and then we'll go on how it stimulates F226 BPAs.
Glucagon inhibits PFK2, reducing the production of fructose 2,6 bisphosphate. This slows glycolysis, helping raise blood sugar levels.
Okay, and then this decrease F26 biphosphate levels will reduce the activation of PFK1.
Exactly. Lower fructose 2,6 bisphosphate means less activation of PFK1, slowing glycolysis and promoting gluconeogenesis.
So confusing, dude. Let's jump to gas exchange and transport, and um, we'll come back to carb metabolism. Okay.
No problem. Gas exchange involves oxygen entering the blood and carbon dioxide leaving it. This happens in the lungs, ensuring cells get oxygen and remove waste.
Of course. And what's the blood gas interface then?
The blood gas interface is where gas exchange occurs, in the alveoli of the lungs. Oxygen moves into the blood, and carbon dioxide moves out across the thin alveolar and capillary walls.
What about Fick's law? What's Fick's law?
Fick's law describes how gas diffuses across a membrane. It depends on factors like surface area, membrane thickness, and the concentration gradient. Basically, it explains how easily gases like oxygen and CO2 move across the alveoli.
And we have the rate of diffusion being proportional to the surface area and to the concentration difference and to the inversely proportional to the thickness of the membrane. Can you explain this a bit?
Sure, Fick's law states that the rate of diffusion increases with a larger surface area and a bigger concentration difference. It decreases if the membrane is thicker. So, thin membrane with a large area and steep gradient makes diffusion faster.
Okay, now I know that it decreases if the membrane is thicker. That makes sense, but why is this true?
A thicker membrane slows diffusion because gases have to travel a longer distance. The more barriers, the harder it is for gases to move quickly across.
Yeah, I mean, I know it empirically. I just wonder how can we prove that I mean, this is kind of stupid question, but in physics, how can we prove this like that? I guess, okay, yeah, well, I guess time. We can do a time experiment or something, right?
Yeah, you can measure diffusion rates across membranes of different thicknesses. By comparing how quickly gases move, you can see the effect of thickness. Physics often relies on experiments to confirm theories.
Yeah. So,
Any other concepts you'd like to explore?
yeah, just give me a second. Let's talk about the alveolar.
Take your time.
Let's talk about the alveolar gas equation.
All right, the alveolar gas equation estimates the partial pressure of oxygen in the alveoli. Factoring in inspired oxygen, carbon dioxide levels, and respiratory quotient. It helps assess how well oxygen is being transferred from the air to the blood.
I don't understand why this is the equation.
It's an equation because it uses variables like oxygen in the air, carbon dioxide in blood, and respiratory factors to calculate the alveolar oxygen pressure. It helps us understand gas exchange efficiency.
Well, can we go a bit into detail, dude? I mean, explain to me why this specifically is the equation, please. Let's
Sure,
let's
it
let's
calculates
break it down to pieces.
all night. It starts with inspired oxygen, subtracts carbon dioxide pressure, adjusted for respiratory quotient, and accounts for water vapor. This
Wait.
gives a precise
So
measure of oxygen available.
Sorry, wait. So we have FiO2, fractional inspired oxygen, and we times it with Pi minus pH2O, right?
We multiply FiO2 by the difference between barometric pressure and water vapor pressure to correct for humidity. This gives the actual oxygen pressure entering the alveoli.
Well, wait a second, dude. Explain to me why this happens.
When air is inhaled, it gets humidified by water vapor in the airways. This reduces the partial pressure of oxygen, so we correct for it in the equation.
Dude, I don't even know what Pi is. You have to explain to us each individual thing. So the full equation is PaO2 equals FiO2 times parenthesis Pi minus pH2O close parenthesis, then minus PaCO2 divided by RQ. And I know none of these. So you have to explain to me by one what this is, why times is true, why.
Got it. Pi is barometric pressure, the pressure exerted by the atmosphere. pH2O is water vapor pressure. We multiply FiO2 by Pi minus pH2O to find the effective oxygen pressure in inspired air.
I don't understand.
No worries, it's a lot of info. Want to keep going with the rest?
Well, dude, no, I want to understand the equation step by step. Break it to as many steps as you want. Let's start by step one. I really need to understand it deeply.
All right, step 1. FiO2 is the fraction of inspired oxygen. It's the percentage of oxygen in the air, about 21% at sea level.
Okay,
We use this to determine how much oxygen is available.
got it. Let's go to step two.
Step 2. Pi, barometric pressure, is the total atmospheric pressure. At sea level, it's about 760 millimeters of mercury. We subtract water vapor pressure, pH2O, around 47 millimeters of mercury, from this to adjust for humidity.
I don't understand PI, to be honest.
Sure. pH2O is the water vapor pressure in the lungs. It accounts for the moisture that air picks up as we breathe. This adjustment helps calculate the effective oxygen pressure.
pH2 I get, I don't get PI though.
Pi is simply the atmospheric pressure around us. At sea level, it's typically 760 millimeters of mercury. We use it to calculate the pressure of oxygen entering the alveoli.
But why do we find the difference?
We find the difference to get the partial pressure of inspired oxygen, correcting for water vapor. This gives a more accurate oxygen value entering the lungs.
Dude, it doesn't just make sense to me. Just doesn't make sense.
It's okay. We can approach it from another angle. What specifically is confusing?
I don't understand. You could persuade me that all these symbols can be switched around.
I get it. The equation structure helps us understand how each factor influences oxygen pressure. We can break it down more if you'd like.
Yeah, why do we minus the PaCO2 divided by the RQ?
We subtract PaCO2 divided by RQ to account for carbon dioxide's effect on alveolar oxygen. RQ, or respiratory quotient, represents the ratio of CO2 produced to O2 consumed. This correction ensures accurate oxygen pressure.
Tell me more about this.
RQ typically ranges from 0.7 fat metabolism to 1.0 carb metabolism.
What
It adjusts the impact
what's
of
the what is respiratory quotient? What is it?
The respiratory quotient is the ratio of CO2 produced to O2 consumed during metabolism. It varies with different nutrients. Lower for fats, higher for carbs. It helps adjust the alveolar gas equation for metabolic differences.
So it's the oxygen consumed and CO2 produced.
Exactly. RQ reflects the balance between oxygen used and carbon dioxide released during metabolism. It's key for fine-tuning the alveolar gas equation.
And how much do we want?
RQ varies, typically around 0.8 for mixed diets. It depends on the proportion of fats, carbs, and proteins being metabolized.
So why do we times the fractional inspired oxygen with the difference between atmospheric pressure and vapor pressure?
We do this to get the effective oxygen pressure entering the lungs. The difference accounts for water vapor, which reduces the total pressure available for oxygen.
Okay. And And so, but why time it with FiO2 and not add it or divide it or something?
Multiplying by FiO2 gives the partial pressure of oxygen specifically. It is how we determine the actual amount of oxygen available for gas exchange.
And why do we minus PaCO2 divide by RQ? Why do we divide RQ with CO2?
We divide by RQ to adjust the CO2's impact based on metabolism. This correction ensures accurate oxygen pressure in the alveoli.
All right, let's talk a bit about the AA gradient then.
Sure, the AA gradient measures the difference between alveolar oxygen, PaO2, and arterial oxygen, PaO2. It helps assess how well oxygen moves from the lungs to the blood.
How? It's because, explain to me. So if we have a steep AA gradient, what does this mean?
A steep AA gradient suggests a problem with oxygen transfer, like lung disease. It means there's a bigger difference between the oxygen in the alveoli and what's reaching the blood.
So if there's a big difference, it means there's something wrong with the with things.
Yes, it indicates an issue like impaired gas exchange, possibly from conditions like pneumonia or pulmonary embolism.
Interesting.
Exactly. It's a useful diagnostic tool.
Sorry. So I understand that the weight of fluid in the pleural cavity is greater at the base because of gravity, yeah?
Yes, gravity causes more fluid to accumulate at the lung bases, which can affect breathing and gas exchange.
So why does it result to the alveoli being less expanded and having a higher compliance at the base?
At the base, the alveoli are less expanded due to gravity's pressure. This makes them more compliant, meaning they can expand more easily with each breath.
Can you explain this a bit more?
Sure, because of gravity, alveoli at the base are slightly compressed. This means they can fill with more air during inhalation, making them more compliant.
Why? Why? Sir. Guys, he's not with us anymore. Let's ask him again. Ask it. All right. Hello, sir. Hello. Technical problem, guys. Technical problem.
No worries, take your time.
It's back. All right. So let's continue our conversation. So I was wondering why we have the alveoli having higher compliance at the base and less expanded. It doesn't make sense to me.
It can seem odd, but it's due to gravity. The weight of lung tissue compresses the base, making those alveoli start smaller. This smaller size makes them more compliant and able to expand more during inhalation.
So I don't understand why this makes them more compliant.
More compliant means they can expand more easily. Since the base alveoli are initially smaller, they have more room to fill with air, making them more flexible or compliant.
Let's talk about ventilation perfusion mismatch.
Sure, ventilation perfusion mismatch happens when airflow, ventilation, and blood flow, perfusion, aren't evenly matched in the lungs, reducing efficient gas exchange. This can lead to lower oxygen levels.
I don't understand. So wait, so what? So what's the difference between ventilation? Okay, perfusion mismatch and AA gradient.
Ventilation perfusion mismatch refers to uneven air and blood flow. The AA gradient measures the difference between alveolar and arterial oxygen. A mismatch can contribute to a higher AA gradient, indicating poor oxygen transfer.
Hmm, interesting. Okay, listen, let's go back to regulation of carb metabolism, and I wanna start from the beginning. So the glycolytic pathway. Let's break it down to 10 steps. Step one.
All right, step 1. Glycolysis begins with glucose. An enzyme called hexokinase adds a phosphate to glucose, forming glucose 6 phosphate, G6P. This traps glucose inside the cell.
Question: why is hexokinase the enzyme that's ideal for this?
Hexokinase is ideal because it's highly specific for glucose and works efficiently at normal cellular glucose levels.
All right,
It ensures glucose.
cool. Let's go to step two.
G6P is converted to fructose 6 phosphate, F6P, by an enzyme called phosphoglucose isomerase. This rearranges the molecule to prepare it for the next step in glycolysis.
So why is glucose phosphate isomerase the ideal one to go from G6P to F6P?
Phosphoglucose isomerase is efficient and specific for this step. It facilitates the rearrangement smoothly, ensuring the glycolytic pathway progresses efficiently.
And then how come PFK1? It just seems to me like these enzymes are random. Like, I'm sure they're not, but I can't figure out why PFK1 is ideal to go from F6P to F16P2 fructose 1,6 biphosphate or whatever.
EFK1 is crucial because it regulates glycolysis. It's sensitive to the cell's energy status, speeding up or slowing down based on ATP levels, making it perfect for controlling this key step.
Huh, interesting. Okay, and then it goes to DHAP or Gly3P. Why are these the ideal ones?
DHAP and G3P are ideal because they provide 2 3-carbon molecules, ensuring efficient ATP production. This split maximizes energy extraction from glucose.
Elaborate a bit more. Instead of explaining what's going on, explain why it's going on.
Sure, splitting into DHAP and G3P ensures the cell can produce more ATP and NADH. This dual pathway boosts energy yield from one glucose molecule, making the process efficient.
Interesting. Okay, I'm gonna take guys. So and let's go a bit more, and then I'm gonna change topics. I might do a new live stream for the new topic. So then Gly3P becomes 1,3-DPG. How come?
G3P is oxidized and phosphorylated to 1,3-BPG. This step generates NADH, which is crucial for later ATP production in the electron transport chain.
Interesting. And then it becomes 3-PG.
Exactly, 1,3-BPG is converted to 3-PG.
All right, guys. So very short episode. It was kind of a test drive for me to see how live works too. Hopefully the audio isn't too bad. I'm gonna try to make it better. I'm kind of learning about glycolysis and it's going very well. But hopefully it's gonna become better. I'll put more hours into it. And yeah, I'm gonna do Truman show special now. So yeah, join me on that. See you guys. ChatGPT, say goodbye.
Sounds great. Good luck with the next live stream. Bye, everyone.